In Objective 4, we're going to be looking at the idea of localization of function. In other words, can we ascribe particular functions, can we determine what functions a particular part of the brain has, what a particular part of the brain does? Can we make a one-to-one correlation between what the brain is doing and what part of the brain is doing that one thing? You'll see that the answer, as always, is it's complicated. To a first approximation, yes, but then it gets complicated beyond that first approximation. So the idea of localization of function was really strongly supported by a lot of evidence, a lot of different lines of evidence during the 19th century, during the 1800s. And so at the end of that process, in the beginning of the 20th century, the 1900s, a German anatomist named Corbinian Brodmann, actually a histologist looking at the microscopic structure of tissue, came up with a map which is named after him. This is Brodmann's map of different areas. And anytime you hear an area referred to, an area of the brain referred to by number, like area 17 for visual cortex, then that's coming from Brodmann's map. And we'll take a quick look at that here a little bit later on in this objective. But that's the idea of localization of function, is that one part of the brain is responsible for one exact function. A lot of support for this came from a really horrible accident which happened to exactly one person in human history. Thank goodness it isn't something that could happen very commonly. This is a man by the name of Phineas Gage, and he's working on the railroad outside of Cavendish, Vermont, on the 13th of September of 1848. And so he's helping to build the railroad. He's the foreman of a crew. His job is to help the crew in this way, is that another member of the crew is going to drill a hole in the rock. These holes had to be drilled by hand. They didn't have machine tools or power tools in those days, and so they would do this stuff by hand. It's a pretty painstaking process. Then when they got a good hole going, then they would put gunpowder down in the hole and a fuse on top of the gunpowder. And then there's a person who's tasked with pouring sand into the hole, and this is because the rock that they're working in is flint, and when flint and steel come together you get a spark, and when sparks encounter gunpowder they make it explode, and you don't want that to happen. So then the fuse was already in there, of course, underneath the sand, and so you go and with the gunpowder and the fuse underneath the sand, and so you go hide behind some other rocks a long way away and then blow things up, right? This is before the invention of dynamite, which is another story entirely, but the invention of dynamite made it possible so that you could transport explosives from one place to another, but in those days all you could do was pour gunpowder in the hole. So the gunpowder went off prematurely. We don't know why, but this drove a steel rod about 1.25 inches in diameter and about 4 feet long with one pointy end, which went through Phineas Gage's cheek and eye and out the top of his head and clattered on the rocks a few hundred feet away. They recovered it, and that's what he's holding there in this photograph here, is the bloody rod that went through his face. You can see the damage to his facial features here in this daguerreotype as well. This is a reconstruction of the path that the rod must have taken through his brain. This is based on the looking at the fracture in the skull. As you can imagine, something moving with this much momentum, that much speed, and that much mass was able to act like a bullet and just vaporize everything in its path. And so it's pretty easy to, the bone didn't shatter very much, and so it's pretty easy to figure out where the entry wound and the exit wound was for this. And this is a reconstruction of that, and you can see the part of the brain that Phineas Gage lost. So what happened in the accident was that his personality changed, but not much else. And so that meant that personality must be resident, or we believe that mostly rightly that personality is resident in this area of the brain which has been destroyed. Similarly, in the 1870s, Paul Broca looked at first an index patient and then a number of other patients who had a disease which we call aphasia. That's the inability to speak. So aphasia here with an S means speaking, and A is not. And so these patients suffered strokes, and after the stroke they were unable to speak. Even today, that's a fairly common result of having a massive stroke is losing the ability to speak, what we call aphasia. The most common kind is called Broca's aphasia now, in Paul Broca's honor. And so Paul Broca made the association between this one area of the brain, which is now called Broca's area in his honor, this one area of the brain, and the ability to produce symbolic language, not just speech with your mouth, but also being able to write with your hand. So any kind of turning spoken language, for example, into symbolic language and coming up with especially coming up with the appropriate response in symbolic language is the activity as due to the activity in Broca's area, and if Broca's area is damaged, then the patient can't do that. It turns out there's a dominant and a non-dominant hemisphere, and so Broca also discovered that for 90-some-odd% of people, it varies between different cultures and at different times and how you measure it, but 90-ish% of people have the localization of language in the left hemisphere and tend to be right-handed, and then a minority of people have the localization for speech in the right hemisphere, and they tend to be there is a higher proportion of left-handed people in that population. It doesn't track exactly. So you can see here in experiments in the dog, and you can notice they're just using any animal that they can get. They're not really thinking rationally about what animals would be best for what experiments. But Fritsch and Hitzig, rather, in the dog, or Ferrier in the monkey, showed that there was a particular area of the brain in the dog or the monkey that if you passed electric current into it, it would cause movement. And so one specific kind of movement associated part of the brain. As a result of the Russo-Japanese War of 1905, and you may not have even known that Russia and Japan went to war at some point in the early 20th century, but they did. And that was one of the first wars that used high-speed rifling, that used high-speed bullets. And that meant that instead of like mini balls during the American Civil War that would go into your head and then all the energy would stay there and vaporize the brains right there inside of your skull, and of course that would cause immediate death. Now we have through-and-through injuries for the first time. We have injuries that have an entry point and an exit point, just like we had with Phineas Gage, but a much smaller injury. And so Inoue, a Japanese ophthalmologist who was studying his patients that had war wounds and had been rendered blind as a result of those war wounds, was able to map out quite precisely the visual cortex in humans by mapping out the location of those different lesions. Now that brings us to Corbini and Brodmann's map, okay, published in 1909. This image is probably familiar to you. You've probably seen it somewhere. And these numbers correspond to different functional areas of the brain. So he gave them these numbers based on their microscopic appearance, what we call cytoarchitectonics, which is the arrangement of nerve cells. What's the shape of the nerve cells? What's the size of the nerve cells? How are they arranged? And he used that to come up with these different areas. And it turns out that those things are, as we'll see later on in the course, are really, really highly correlated with brain function and with the function of individual neurons. So we need to get it drilled into our head that the function of neurons depends on a neuron shape, that anatomy and physiology in all cases are inextricably linked. And the anatomy of the brain and the physiology of the brain are linked together with one another in this microscopic way as well. So we can define microscopic regions that turn out to be different functional regions. We're going to be making reference to this map throughout the rest of the course and throughout any career that you have in neuroscience. These are commonly used reference points. Okay, so now we're all set up to find out what is the location of memory Okay, learning and memory. So where are memories stored? So if I have, for example, a grandmother, and I have two of them, but there's one in particular I'm thinking of. We call her Tot. And if I'm thinking of Tot, and then there must be a cell in my brain that neuroscientists actually called the grandmother cell when they were looking for this. There must be a cell in the brain that contains a representation of my grandmother. And so it's just sitting right there, and anytime it's turned on, then I have an image of my grandmother, right? Very logical and an obvious kind of way of organizing things, also just dead wrong. That's not the way it works, and the way it works we're going to have to save for later on. Carl Lashley of Harvard and others were looking for the location of memory in the brain, and they made the critical rookie mistake of calling it by a name before they actually found it. So they would tell people what they were looking for, and what they were looking for, which is something called the engram, which is the physical location of a memory trace. And you've probably seen this in cartoons, you've probably seen this in movies, you probably are really familiar with this, you had no idea where it came from. This is where we get rats and mice running in mazes to get a piece of cheese as a reward. This is what Lashley was doing. Can they remember where the cheese was located? And so then what he would do is, once the mouse or the rat had learned, usually rats, had learned where the cheese was located, then he would go in and do surgery on the brains and remove areas of the cortex. If he found an area that when he removed, made them forget where the cheese was, then he had shown where the engram was. However, these were a complete and total failure. There is no engram. Engram no está aquí. Okay, it's not there at all. It turns out that it's not a particular location, that it's a process. And this process is found all over the cortex. We're going to study this process later on. If you want to skip ahead to the chapter on modifiable synapses, that's where we're talking about it. But it's a particular type of synapse which is found all over the brain. It's not a particular location. And so if I have a visual memory of my grandmother, it's probably stored in my visual cortex. If I have a auditory memory of my grandmother singing, then it's probably in my auditory cortex. And if I have a tactile memory of my grandmother's touch, then it's in my somatosensory cortex. So it's in the cortex that corresponds to the sensory system that initially recorded the information in the first place. And you'll notice that I keep saying cortex over and over again. It turns out that there's another important concept embedded here, which is that cortex equals consciousness. You'll hear me say this over and over again in these recordings, and if you have me in the classroom, you'll hear me say it over and over again in the classroom, because it's really important. Cortex equals consciousness. So if we're going to be consciously aware of something, that's because of a pattern of electrical and chemical activity in our cortex. Visual cortex for visual activity, auditory cortex for auditory activity, and so on. Now in the first half of the 20th century, from about 1900 to 1950, a surgeon named Wilder Penfield, now there's a great name, just like Corbini and Brodmann. There were giants in the earth in those days, and they had much better names as neuroscientists than prosaic names like Jim Hutchins or whatever. But Wilder Penfield decided to do something which today would be impossible, because you could not get informed consent to do this to a patient. He was doing good for these patients. He was removing their brain tumors. He was the only person in the world who could, so that part was good. But the part that was bad was he decided to do experiments on their brain while he was in there, and ostensibly so that he wouldn't cut out a part of cortex that was important. But as you can see in just a minute, the parts where they probed turned out to be pretty far afield from where the actual lesion was. And so here's numbered tags that they put in the brain. The brain that you see over here is upside down to you. So this would be the top of the patient's head here. This would be the patient's jaw down in this region here. And so you can see they put these numbered tags on the brain, and these are sterile, of course. They don't want to kill the patient quite that quickly. And so then they would stimulate with an electrical device called a thyristor. They would stimulate with different voltages in different areas of the brain. And so the number tags correspond to these. Let me read you a few of these. So if we stimulate at number 15, which is this place right here, then we get a sensation in the volar surface of the forearm with a slight movement of the thumb. So mostly sensation, but a little bit of movement of the thumb, and a sensation in that part of the forearm. And in that way, we can map out what's called a homunculus, which is Latin for little man, a map of the entire cortex, both for motor cortex, for movement of the left forearm and hand, and number 34, which is a little bit forward. This is towards the front here, because this is frontal cortex. A marks the location of the tumor, by the way. And so here, this is now known to be somatosensory cortex, where you feel things. So if you look at the entries in the diary that correspond to these numbers, you'll see that there's sensations. If we're in this area here, this corresponds to movement. And so there's a movement homunculus just in front of, there's a movement map on the surface of the brain just in front of the sensory map of the brain. And so this is showing that process. The numbers as they appear to the surgeon are upside down, but this is so you can sample Brodmann's areas here. So here's Brodmann's 17, Brodmann's 18, Brodmann's 5B, and so forth. So those are the right side up numbers. The upside down numbers are the numbered tags that they were putting on the surface of the brain. Here, A is where the tumor is located. You can see, even though this is upside down, that this is marked as the tumor. And so you can see that these sterile tags and the stimulation of the brain was happening in areas that were pretty far away from the tumor that they were operating on. But before about 1950 coincidentally happens to be after the Second World War, was the time when our modern conception of medical ethics kind of came about. And so that's why this would not be permitted any longer. Although things that you would think of that are even worse than this that we do today are permitted. I'll leave that to you to do as an experiment and something to look up. If you're interested, and even write me a textbook chapter on it would be absolutely great if you wanted to do that.